Right, so the first main enzyme is helicase, and helicase's job is to unwind the DNA. Remember, they're stuck together. So it almost acts like a doorstop, and it's represented here by the triangle. And it keeps these complementary bases from sticking together. The second main enzyme that you're going to be held responsible is called DNA polymerase. His job is to add the nucleotides that are complementary to the original DNA strand. So if you look here, DNA polymerase is represented by this blue circle, and it's basically reading the DNA and bringing the proper nucleotide, the complementary nucleotide, to each of the original DNA strands. All right, and the third step is the DNA polymerase is released, and we have two DNA molecules that are identical to the original strand. Again, it's semi-conservative because one is the original, one strand, and the other strand is new. Okay, so let's look at these main players one more time. This is what you will be held responsible for. You need to understand that this process of DNA replication requires certain enzymes, okay? And you need to know their names. So, helicase, again, uncoils, and it separates it, all right? DNA polymerase inserts the nucleotides. It also proofreads and makes sure that the right nucleotides are added. That's a very important job. And then this guy we haven't talked a lot about, ligase. He binds the fragments, but you're right now thinking, what fragments? Well, we're going to talk about those fragments. Okay, now this is worded in your tests in a way that scares a lot of you guys. Okay, so we said DNA polymerase brings the nucleotides, right? That's his job to the complementary strand of DNA. Now, what scares people is the wording that you'll see on the test, and you've got to start to think like a scientist. DNA polymerase catalyzes. We've talked about the word catalyst a bunch by the time we've gotten to this unit. Catalyzes means to initiate, right? It starts it up. If you have a catalyst in a chemical experiment, it's the thing that lowers the activation energy. It allows the chemical reaction to happen. So it catalyzes the addition of nucleotides to form a complementary strand of DNA. So that's a fancy way of talking about the job of DNA polymerase that I'm going to go ahead and hold you accountable for, because you need to talk like a scientist. All right. Now, this is where we're getting a little confusing. And if you watch videos, this might get confusing for you. I've got the amoeba sister video I'd like for you all to watch, and there's another video. You just need to know for this class this year that there's such a thing as a leading and a lagging strand when we make DNA. And that's all that you need to know, that the leading strand is right here, and it's just one continuous strand that goes towards this replication fork. Okay? The lagging strand is created in fragments. And I'm going to explain why it's created in fragments, but you don't have to answer the why on your test. What you need to know for your test is right here on this PowerPoint. A leading strand is on one side when DNA synthesis occurs, and it's a continuous. A lagging strand is on the other side, and it's in fragments, okay? These fragments are called Okazaki fragments, and they're glued together by DNA ligase. That was another enzyme that I want you to understand. He glues these fragments together. Now, that's all you have to know for this test. For some of you, though, you might want to know more, especially if you're going to be in AP Bio, okay? You will not be tested on this, I repeat. But the reason we have a leading and a lagging strand is we talk about the 3' and the 5' of DNA. We say it's anti-parallel. One side is always a 5' and one side is always a 3'. I explained why, I believe, earlier, but You just have to know you're always going to have a 5 and a 3 prime. So every DNA strand, if you look at this blue guide, there's a 3 and a 5 prime, just like we have a head and a tail, or a head and a bottom, right? Well, DNA, we can make 5 the head and 3 the bottom, and 3's the bottom and 5's the head. You don't see people walking around without a head or walking around without a bottom. You gotta have both, okay? Now, here's the rule with DNA. It's anti-parallel, meaning if this is a 3 prime, the 5 prime has to be on this end next to the 3 prime. So DNA likes to line up head to tail with the other strand. So this comes into play when we try to replicate DNA, because if we've got the 3 prime end here, the head that has to end up over here is a 5, and a 3 has to be on this end. Well, DNA polymerase is a drama queen and will only add to the 3 prime end. So it can't add to this end, so we end up making fragments. Now, if I lost you there, do not worry. You're going to watch a bunch of videos, and if you're still lost, don't worry. We're going to go over it next year, and you're not going to be held accountable on your test for this concept. You just need to know that DNA synthesis occurs, and it has 2 types of strand, a leading and a lagging, and the leading is the one that occurs continuously. All right, so here we have DNA replication. This is just a quick summary. The parent DNA makes 2 exact copies of DNA. This occurs in cells before mitosis. Remember the S phase. That's when this is happening, okay, of the cell cycle. So the summary of DNA replication: DNA has the ability to make a copy of itself, and it does so through DNA replication. It unfolds, you have 2 strands exposed. That enzyme is called helicase. It's not on there, but just remember. And then we have a nucleotide, the base pairing, which occurs through the enzyme DNA polymerase. The process moves down, and once completed, we have 2 identical strands, okay? Now, you have to know that we have this thing called Okazaki fragments, which is in the lagging strand. And the lagging strand is put together by DNA ligase. Okay, so let's look at this one more time. The replication steps. First, you have initiation. We find a spot, okay? Then you have elongation, and we start adding the bases, and then we have termination. Now, it is interesting with DNA replication, and there's lots of videos that you're going to find in your notes. Go ahead and look at those videos. I've only included a couple on the Google Classroom link, but you can easily follow them if you're following the notes through the Google Slides. What enzyme unzips DNA? If you want to test yourself, pause, read these, answer them, and then I'm going to go ahead and answer them right now. The enzyme that unzips DNA is helicase. What catalyzes the addition of nucleotides to form a complementary strand of DNA? That's going to be DNA polymerase. And why do we call this process semi-conservative? Because you use one of the original strand to create one of the new strands. Now, here's a fun thing. If you were going to do DNA replication, what would be the complementary strand that would occur? This is just where you get to apply your base pairing knowledge, okay? So you're going to end up, and I'll write it down, with G, because G binds with C, A, T, C, A-T, G, A, okay? And I have circled, kind of, sorry, it got a little messed up. I have circled, and I'm going to put it on the screen right now. Here is the strand that you should have come up with. This was the one that you should have found the coding for, okay? All right. Now, errors can happen in DNA replication. That's where we get these mutations. Now remember, not all mutations are bad, And DNA polymerase is responsible. This DNA polymerase is has a man of many talents. And it proofreads the new DNA and catches any mistakes. But mistakes can be made, and they also cannot get caught by DNA polymerase. And that's where we have things like cancer. But it's not always bad. We can also have sources of genetic variation, mutations. And again, mutations aren't always bad. And then you can have crazy things that happen with mutations where you have frame shifts, substitutions. We'll go into that later more. But this is where you might end up with like a disease called sickle cell anemia, where your red blood cells are sickle shaped, all because one amino acid was substituted incorrectly. Okay? All right. Now this is interesting. Errors occur in 1 in 10 billion nucleotides. Humans have 3 billion base pairs in their DNA. So we're pretty dang efficient at this process. When mismatches occur, DNA polymerase can proofread it, and it's sort of like if you proofread it and you delete it or you do whatever, it might cut it out and it's going to repair it, okay? But sometimes that repair doesn't happen, and that's where you get things like the BRCA gene or any kinds of cancers. And there is, at the end of this, there's a video I'd love for you to watch that discusses the BRCA genes and cancers and how they happen because of these DNA sequencing errors. All right? Okay. I think you guys are prepared to do the worksheet that goes over DNA replication. It asks a lot of detailed questions. I would request that you don't do that worksheet until you have read it, well, I would say watched all the videos, and if you're still confused, read the pages on the book that I have included dealing with DNA replication, and then do the worksheet. There's lots of material that will help you answer the questions that are on that worksheet. Okay, thank you guys so much.